Welcome everyone to this, it's the third or the fourth NCIRS seminar, and we're privileged today to have both an international topic and a speaker of international renown in Professor Ben Marais in this area of global tuberculosis with a particular focus on BCG. Ben's a paediatric infectious diseases specialist who has a particular interest in global health and particularly with reference to that in TB, both in childhood TB more generally and also in multi-drug resistant tuberculosis. He's been one of the key members of the NHMRC Centre for Research Excellence in Tuberculosis Control and has also played a leading role in TB and multi-resistant TB control efforts in the region, and in addition to his university appointment is Deputy Director of the Marie Bacille Institute. So we look forward very much to hearing from Ben on the topic of tuberculosis in children and the role of BCG in control. Thank you.
I should just thank Peter for the opportunity to talk to you and for the interest that NCIRS has shown in BCG, because it has been one of the orphan vaccines, not only in Australia, but globally. And I'm going to leave most of the BCG discussions over to Frank. So I will just briefly provide a background introduction to the global TB epidemic and how children are affected, and share some data from Sydney and close neighbour countries. So briefly, for those of you who are not working in the TB field, I think it's now believed that TB is probably the archetypical human hominid pathogen. And there's very good data to show from archaeological evidence that the emergence of TB as a respiratory pathogen in humans is probably related to the fact that we are the only species that have controlled fire. I'll get back to that in a minute. For a long time, we've believed that TB have evolved from animal strains of mycobacteria, but when the first genetic analysis became available in the early 2000s, it was clear that TB seemed to be the ancestral strain of all our animal strains, and that human TB probably, or that TB as a respiratory pathogen probably emerged in humans first. More detailed genetic analysis in recent years have shown that there's essentially 7 big lineages around the world. 3 of them on the left-hand side are currently the major drivers of the global epidemic. The best available thesis is that all these strains emerged in Africa. They migrated with human populations all across the globe, which explains their current geographic distribution, but obviously there's a lot of mixing happening with European colonization of different parts of the world. TB strains have moved with those populations, and we can fairly accurately retrace the colonization history of any particular country by just looking at the TB strain distributions in that country. If we focus on the current global burden of tuberculosis, in 2015, TB was the #1 infectious disease killer on the planet, killing more people than HIV and malaria combined. Obviously, it's an ancient foe. Everyone have thought that we won't be talking about TB in 2017, but it still killed nearly 2 million people. And we have been becoming increasingly aware of the burden of TB in children, especially young children. Current best estimates are that there are annually around 1 million children who develop TB, with around 100,000 deaths in children who are HIV uninfected due to tuberculosis. Obviously, the disease burden isn't spread equally. TB has always been a reflector of social inequality, and if we see where the global TB burden is distributed, you can see that it's in sub-Saharan Africa, and in the Asian Pacific region. The 2015 Global TB report really illustrated the local phenomenon very clearly, I think, where you can see that the 3 countries that are most heavily impacted or have the highest case numbers now are China, India and Indonesia, with India being an outlier with by far the highest burden. Indonesia has moved into second spot recently because Indonesia has conducted a TB prevalence survey, which showed that estimates of TB disease, national estimates of TB disease, probably underestimated the TB burden by around 100%. So they literally doubled their TB rate following an extensive national TB prevalence survey. Partly it's because TB in many of these countries are found in socially disadvantaged populations who poorly access healthcare. So often when you do a formal prevalence survey, you get an unpleasant surprise at the number of cases that are undetected in your population. You can also see, if you look at absolute numbers, so numbers per population, then sub-Saharan Africa and countries that are suffering the dual epidemic of TB and HIV have the highest disease burdens. But Timor-Leste and Papua New Guinea is not included on this slide because it didn't have accurate enough numbers. But current estimates are that Papua New Guinea are also in excess of 500 per 100,000. So you can see both Kiribati, Timor-Leste, and Papua New Guinea, Cambodia, Myanmar in that very high disease burden range. If we talk about TB, often people working in developed country settings like Australia will say, but we hardly ever see children with TB, and it's true, our case rates are low, although we do see, and I'll give you a brief overview at the end, but people's experience are heavily influenced by the environment within which they work. This graph, I think, illustrates the 2 most important epidemiological characteristics. Is if you work in a low burden environment where the disease rates are low, so less than 1 per 100,000, or in that order of 1 per 100,000, which we find in 10 per 100,000, which we find in Australia, and you've got a mature population demographic, you essentially have few children in contact with few TB cases, you get very low burdens of pediatric TB, and pediatric TB cases on average in developed countries are about 5% or less of the total TB burden. But if you go to a place like Timor-Leste or Papua New Guinea or South Africa, where you've got disease rates close to 1,000 per 100,000, and you've got a developing country population pyramid with lots of kids under 15, a place like Timor-Leste or Papua New Guinea, more than 50% of the population are less than 15 years of age. You get lots of exposure of young, vulnerable children with high disease rates, and in those settings that have accurate data, estimates are that in excess of 30% of the TB disease burden occurs in children. We've had very poor estimates of TB mortality in children under 5. The red light was really lit when there was an autopsy study done in Zambia. Professor Shintu was the leading academic light in pediatrics in Zambia, and he was seeing a lot of children under 5 dying and not responding to normal antibiotic therapy. At the time, they believed that all of this was due to HIV, so they embarked on a 2-year autopsy study where all children dying from respiratory disease in the Zambian National Hospital was autopsied, or they were able to autopsy in excess of 75% of children, so remarkably high rates of parental consent. But they showed that although TB, well, first of all, that about half the children who died, in fact, did have HIV, and PCP was the leading cause of death in that group. But TB was common in both groups, both in HIV-infected and uninfected children. It was, in fact, the second leading cause of death after bacterial pneumonia. And in all those children dying from TB on autopsy, they were reported as pneumonia deaths, not as TB deaths. So it's completely below the radar screen in many countries, and recent estimates are that TB are among the top 5 causes of death in TB endemic countries with uncontrolled transmission. In many of these countries also, the relative importance of TB is increasing as vaccination programs are being rolled out. So in South Africa, for instance, where we've had very effective hip and pneumococcal vaccine programs for the last 10 years or so, TB has suddenly become the most common cause of meningitis and has remained so. And I assume the same will happen in places like PNG and East Timor if we're able to roll out vaccines against common preventable diseases more effectively. The relative contribution of TB, and more so in the absence of BCG, will increase. There has been recent estimates. There's been a recent Lancet Infectious Diseases publication looking at the mortality contribution of TB in children under 15, and there should be a Lancet Global Health publication on the contribution in children under 5 coming out soon. So just looking at what do we know about childhood tuberculosis and how do children present clinically, we know that the MTB is taken up by pulmonary macrophages, and from there it drains to the regional lymph node, which is part of the primary infection complex. That's where T-cell priming happens, and then you get clonal expansion, and you get granuloma formation at the site of entry, which often leads to containment, but it can also lead to poor containment or disease progression. So the main drivers of disease progression are either inadequate immunity, which leads to inadequate containment, or you have an hyperaggressive response, which leads to cavity formation and lung destruction, which is what we see mostly in adults. In children, and especially children under 2 with immature immune systems, most of the disease spectrum is skewed towards poor containment and disseminated disease, rather than cavity formation, which is typical in adults. So a very important distinction to make, which we don't always make in infectious diseases, but in TB it's very important, is that infection doesn't equal disease. So the major transitions in TB is exposure. We know Robert Koch showed you can't get TB without TB exposure or TB infection. So not all children exposed become infected. There's very clear variables related to the risk of aerosol transmission, which determines your risk of infection and your closeness of contact with an infectious case. But then the real important distinction is between infection and clinical disease, which can be problematic in children. So we previously reviewed all the studies that were conducted prior to the availability of chemotherapy, so prior to 1950, to describe the natural history of tuberculosis in children. So prior to 1950, TB was still a big problem in Europe, even in the US. There were big child sanatoria. There was a place called Highwood in London, Bellevue in New York, a big sanatoria in Scandinavia. And there were meticulous clinicians who recorded exactly what happened to these children in the absence of treatment, just giving them sunshine and good nutrition. But what they showed was, following documented TB infection, which they recorded with a skin test at the time, the highest risk for disease progression occurred in children under 2. And we know that this is very similar to what we see in children with severe immune compromise, HIV-infected children or immune compromise from other conditions. So in those children, the risk of disease progression exceeds 30% and is close to 50%. Those young children also have a very high risk, the red bars, of disease dissemination. As children move into the primary school years, 5 to 10 was in those old studies referred to as the safe years of primary school. Your risk of disease progression is lowest. And then as children move into adolescence, suddenly their risk increases again, but now they have a phenotypic switch towards adult-type disease. They no longer have the typical pediatric forms of disease. The other important determinant of risk in these old studies, we often get taught that TB is a disease of reactivation. So you get primary infection, then you have a long latency period, and then you get reactivation some point in the future. This is true if you live in Australia or in Japan, where we see most of the TB in older age groups, people that were infected long ago and now have immune senescence or immune suppression. But in high burden settings and in these old studies, the bulk of disease, and in children, more than 95% of disease occurs within the first 12 months after infection. So the window of risk is really that first year. In adults, that window of risk extends to 2 to 5 years. Even though you can get late reactivation, the majority of disease occur fairly short after infection. The other important fact is that infection is not a single event. If you live in a high-burden country, if you live in a slum in Calcutta, you may get infected 20 times a year. You may get infected every evening that you sleep in an environment where many other people are coughing. So infection is not a single event. Reinfection, and now we can record reinfection with new genotypic data, occurs commonly in high-burden settings. So there's a whole spectrum of disease manifestations that can occur in the lung of children, but also lots of extrapulmonary manifestations. I don't have time to go through all of them. There is a review that you can look at that summarizes some of these, and they are all linked to typical X-ray pictures that we see in children. I suppose the bottom line is that unlike adults where we can say this is what TB looks like, it's a cavity in the lung apices, in children on the whole, there's a whole spectrum of disease that you should consider. And the majority of children present with hilar lymphadenopathy as the main disease manifestation. Obviously, the most concerning manifestation that we want to prevent is disseminated forms of disease, miliary disease, and TB meningitis. And this is the one disease manifestation where we know that BCG offers protective effect, and where all the Cochrane reviews, and I'm sure Frank will share some of that, shows consistent protection. There hasn't been consistent protection shown against adult-type disease and breaking the epidemic cycle. But protecting children against severe forms of disease, we've got good evidence that BCG is efficacious. When I mentioned those different strains spreading all around the world, I didn't emphasize the one strain, the Beijing strain or lineage 2, which we find in East Asia. That is the one strain for which there is evidence to suggest that BCG provides suboptimal protection. So it's a strain that may have evolved partially to escape the protection provided by BCG, but there's a lot more to be done to confirm that. So briefly on TB diagnosis, you may all be aware that we've got TB infection tests, either the skin test or the IGRAS or T-spot TB, which doesn't differentiate infection from disease. They are still problematic because they are not optimally sensitive or specific, although the IGRA is far more specific than the skin test. So the quantiferon is the test that we all use in the hospital, but just so that you're aware that there's a new test, the quantiferon TB Plus, which will probably replace the quantiferon very soon because it comes at a higher price and it's currently marketed as the only available alternative. The underlying rationale for the quantiferon plus was we want to optimize the sensitivity because quantiferon has suboptimal sensitivity, especially in young children. So they've included a new set of antigens that are short peptides that are supposed to stimulate CD8 cells, while the previous tests were focused on CD4 gamma interferon production. However, the preliminary evaluation of this new test have shown essentially no additional yield. And if you can see that in the antigen pools here, and pool 2 is the current antigens that we use, you essentially were able to detect all the infected cases. The CD8 specific antigens haven't added a lot of sensitivity, but this was just the first preliminary study. Hopefully follow-up data is more convincing. So then there's other new approaches. Now, looking at other cytokines beyond gamma interferon, and there was a very interesting study from Melbourne published in 2015 that showed that if you look at a whole array of cytokines, you may get a more sensitive and a more specific readout. But this was done in a very small TB cohort in Melbourne. It still needs to be validated in a high TB burden setting, and they're currently busy with that. Alternatively, there's the option of saying we don't want to measure any T cell that produce gamma interferon. We only want to measure those specific T cells that are associated with disease or infection. And there are new techniques for phenotyping the T cell population and then testing the TB-specific reactivity of a specific T cell subpopulation to help you differentiate TB infection from disease. And there are promising tests like the TAM-TB assay that's been published in 2014, but that at the moment is a very complicated test that needs high-end laboratory infrastructure and people are currently working to see if they can simplify and validate those results. And then, like in all diseases, there's this possibility which all of us is hoping for, that there'll be a unique gene expression profile associated with TB, which we can use. And there has been studies that have looked at diseased children versus non-diseased children that have shown that there's a 50-plus gene expression profile that, with a fair amount of accuracy, can identify TB disease. Studies are currently ongoing to try and reduce that down, looking at an expression profile that will look at only 8 to 10 markers, which we could more easily package into a simplified point-of-care test. The problem, though, is that I've shown you the picture of the diversity of disease manifestations. And there's every expectation that the gene expression profile in a child with a pleural effusion of TB will be different to a child with a cavitary lesion to a child with a lymph node disease, because the underlying pathogenesis is different. The hope is that there will be some common expression profile that is that extends across all disease phenotypes. And previous studies haven't had the power to look at specific disease phenotypes, to look at the diagnostic performance in different phenotypic subgroups. And then there's ongoing studies, some of that at the University of Sydney as well, looking at microRNA profiles in the blood. So in kids, obviously, we're very keen to find a sputum-independent diagnostic method, because you can't get sputum in any child under 5 or 7. The group that you're most concerned about are completely inaccessible with our current testing tools. All right, what about the EXPERT, which is a PCR-based test which is now being rolled out, or has been rolled out in most TB endemic countries, and Papua New Guinea, for instance, now has 21 EXPERT machines. So the EXPERT is a very specific test that amplifies MTB DNA. Initial indications are that in children, it does have reasonable sensitivity, but it's no better than culture. In fact, it's about 3/4, so the sensitivity is about 75% that of culture. Culture is already a non-ideal diagnostic tool. It only detects 30 to 60% of kids with TB, depending on their disease manifestation. So we are encouraging people to use EXPERT, but a negative EXPERT doesn't rule out TB, unfortunately. There's a new EXPERT test that's now starting to be rolled out, which has two adjustments. The one is the EXPERT Ultra element. So the Ultra element, they have reconfigured the probes that detect MTB for added sensitivity. And the indication is that the new EXPERT Ultra is as sensitive as culture. In all the lab experiments, it performed equally well as culture. In addition, there's the Omni version, which is the one on the right, which is a little battery-powered machine, PCR machine, which you can take out into the periphery. If you have a little solar panel, you can run your Omni machine off that, and you can do a PCR test that's TB-specific and hopefully sensitive in any location in the world. So those tests are currently being rolled out and being evaluated, and there are also studies planned in Papua New Guinea. So briefly, just to finish up on drug-resistant tuberculosis, because BCG is relevant, it's the one method we have that provides protection irrespective of the drug resistance profile of the organism. INH preventive therapy is a good way of protecting children, but it doesn't protect children exposed to MDR TB, but BCG can. For a long time, we've believed that drug-resistant TB isn't as transmissible as drug-susceptible TB. But the fact that we've seen children with MDR TB, wherever there has been adult cases of MDR TB, has always been a signal that MDR TB is transmitted, because children don't acquire resistance because they don't drink their drugs, like people assumed adults do. Children develop disease because they get infected by someone in their immediate environment. So that's always been the signal that transmission is happening, because in all the countries where we see adult cases, we see pediatric cases whenever there's diagnostic capacity. This is just data from Papua New Guinea, where there's a big outbreak on Daru Island of drug-resistant tuberculosis. And when we genotyped the strains from that epidemic, And this is from mirror typing, but you can see that's essentially one big cluster. So all these strains, the 200 plus cases of MDR-TB on Daru Island, share the same strain, and they've clearly infected each other. And these are adult patients. There are some children, about 20 children, but it's convincing genotypic evidence of transmission. When we look at where this strain that's causing the outbreak in Papua New Guinea is located according to the lineages I've described, it's clear at the top that that green cluster is a modern Beijing lineage. It seems to be unique to Papua New Guinea. It's different to all other Beijing lineages that's been described and was probably introduced into Papua New Guinea some time back. In order to get a good sense of how long has this been ongoing for, because there's been huge friction between Papua New Guinea and Australia around how did this emerge, who's to blame, is the fact that there are now these MDR and XDR strains because Papua New Guinean nationals have had access to TB treatment on the Australian side of the border. Often people access treatment and then disappeared again without completing their treatment. Some of the drugs to which we now have resistance to in Papua New Guinea was never available in Papua New Guinea, so people must have accessed it somewhere else. So when we did the phylogenetic analysis and trying our best to assess the timing of when the different resistance mutations have been acquired, this picture emerged, and you don't have to focus on everything. The main point is just, if you see the first two bullets, that the first resistance mutations against INH and rifampicin seem to have occurred back in the 1960s. So this strain, this resistant strain, has been transmitting and evolving for decades, but it went completely below the radar screen because we were only testing people with sputum microscopy. And then you can see how the strains evolved in the coming years, how new resistance mutations accumulated with the availability of new drugs, and then essentially some of the XDR strains, which now have resistance against most of our first and second line drugs. It's true that access to treatment or poorly supervised treatment on the Australian side of the border may have contributed, but the main MDR clone has been in evolution for many decades already. So this recent paper, I think, presents a nice summary of where do we think in the world do children have MDR-TB? And you can see that the disease burden is heavily skewed towards sub-Saharan Africa and the Asia-Pacific region. You can also argue this is the part of the world where children are in most desperate need of BCG. So lastly, just to briefly talk about the TB disease burden in Australia. In children, in Australia, we don't have very high disease loads. Children on the whole contribute less than 5% of the disease burden seen in Australia, which is mostly seen among immigrant groups. This was a review of all the cases reported to the National TB surveillance group in Canberra over 10 years, and you can see that children following, essentially contributing 5%, that most of them were detected either by contact screening or following passive case detection. So passive case detection means that these children presented with symptoms. So the health system didn't go out to find them like we do with contact screening. They presented to the healthcare system, and most of them following exposure in a country outside Australia, or exposure outside Australia. Unfortunately, national data doesn't record BCG vaccination status at the moment. It was never recorded, and we hope that that's something that could be corrected going forward. Leila Al-Jazidi, who's one of our infectious diseases registrars, is just putting together the experience in the Sydney Children's Health Network, so both Westmead and Sydney Children's Hospital. She's looked at a 2-year period, 2014 and 2015, and identified 23 children that we treated with TB in that time. And you can see that about half of them were born overseas, but nearly all of them had at least one parent born in a high-burden country, indicating a likelihood of travel to TB endemic settings. In fact, half of them reported recent overseas travel, but around half of them were not BCG vaccinated, and many of them did not have access to BCG. If we just briefly look at the disease spectrum that was recorded in those that were BCG vaccinated or non-vaccinated, the main thing is that there's more kids that were non-BCG vaccinated who had severe forms of disease, and that all the meningitis cases occurred amongst children who were not BCG vaccinated. So I'll leave it there and hand over to Frank to talk about BCG and access issues in Australia.
Well, thanks, Ben. I think it'd be good to maybe just see if there are a couple of specific questions about your talk. I'd like to ask you something about your very early slide about TB burden. I mean, is the fact that South Africa is off the graph, to what extent is that due to the fact that there are better medical services better able to detect TB in South Africa, and to what extent is it HIV related?
So Peter, it's been a long-running discussion in South Africa, where I gather you all know I'm from originally. But initially there was a very high rate of TB in the Western Cape. And there was a lot of studies that looked at the genome-wide association of vulnerability in the local population, whether that was a factor. But it turned out that it was probably mostly detection bias, that the Western Cape was this part of the country with the best access to healthcare and the best access to diagnosis. When people have looked at other parts of the country, disease burdens were equally high. But then the disease burden just essentially skyrocketed when HIV prevalence increased. So we had high disease rates to begin with and high rates of transmission, but with the HIV epidemic, and remember in South Africa at present, there's about 30 to 35% of pregnant women presenting to healthcare facilities, state-run healthcare facilities are HIV infected, and the population infection rate is calculated at around 20%, so exceptionally high rates of HIV infection in the community where there's lots of TB transmission happening. So HIV is definitely fueling the epidemic, and about 75 to 80% of all TB cases in South Africa are HIV infected, which has led to huge stigma issues, because anyone going to the TB clinic to get their TB tablets essentially is telling to the community, I'm HIV infected, or that's the interpretation, which has led to huge barriers to TB diagnosis. But despite the high burden in HIV, when we look at children, whether they are HIV infected or not, have high rates of TB, because often in HIV-affected households, where mothers now have good access to antiretroviral treatment, the kids are uninfected, but they're still exposed to HIV-infected adults and family members who've got very high rates of TB. So the TB exposure rates in those households are high whether the child is HIV infected or not, and they are vulnerable whether they are HIV infected or not.
Okay. Well, thanks very much. And so, any other quick questions? Yes, Robert?
The political concerns with treating patients from Papua New Guinea who may not complete their course, how is that resolving now?
Yeah. So, Robert, since 2014, essentially, all the TB clinics that were provided on the Australian side on those outer Torres Strait Islands have been closed. All patients presenting with TB across the border, so there's free movement across that border, the Torres Strait, because that's part of the treaty that Australia signed with Papua New Guinea at the time of independence, are now directed to Daru Island, where DFAT has built a large containment hospital, or a TB hospital. So there's a close to 100-bed TB hospital on Daru Island now, where they do have good isolation facilities, and that's where most of the MDR TB patients are currently cared for. There are still the odd Papua New Guinean national who are accessing TB care in Australia, but that's a very small number, and there's now very good systems of communication. So there's a cross-Torres MDR care group where the clinicians from both sides regularly meet to discuss patients, and before a patient is relocated back to Papua New Guinea, there's proper handing over of that patient, which didn't occur necessarily 5 or 10 years ago.
So Daru Island is part of the solution. It looked like it might have been part of the problem because of the outbreak there.
Well, it's part of the solution in the sense that it provides the services, but it's definitely the focus of transmission. Daru Island has got unique epidemiological factors that makes it an amplifier within the Western Province. So the Western Province of Papua New Guinea is very rural, very, very poor infrastructure. Daru Island is the nominal capital and the administrative capital of the Western Province. It's the only place where people can access banking facilities, for instance. there's lots of payments that are made to people in the Western Province for environmental degradation caused by the big Octedi gold mine, which was Australian owned, it's now Papua New Guinean owned. But those payments, those social payments, happen on Daru Island. So people from all over the Western Province travel to Daru Island 2 to 3 times a year to access these payments, leading to a lot of social mixing, people staying in very close environments, often for a week or two, receiving their grant handouts, and it's just an amplifier for TB transmission.
So grant handouts plus TB. So I think maybe one more question now, and then we'll have some more general questions. So I think Clayton might have been first. Is that right? Yeah.
Thanks, Ben. You showed the graph showing the probability of the risk of developing disease after infection across age groups. What are the reasons for the teenager of 10 to 15 actually having a high probability developing chronic disease compared to say 5 to 9? And are there some confounders in measurement and ascertaining?
So I'm fairly confident that there's no confounding that's explaining this issue, because it's been a consistent observation across many studies and across many years, and even in the absence of treatment. There is a very poorly understood immune switch that happens around adolescence. The interesting feature is that this adolescent or adult type disease manifestations occurs first in girls and then in boys, really mimicking the onset of adolescence, and that the cavities seem to be associated with a more aggressive immune containment, so a T cell switch, maybe more gamma interferon production. We've been trying to interest immunologists in really exploring this immune phenotype switch. We see the same in some animal models, where in guinea pigs, for instance, they are tremendously susceptible to TB, a lot like infants. But rabbits are remarkably resistant, and if rabbits do develop disease, they need a higher infectious dose, and if they develop disease, they develop cavities in their lung, like an adult. But we don't understand the exact mechanisms underlying.
Okay, well, I'm sure we could keep going quite a bit longer on TB, and we will.